In this video, we are going to study about a very important test, enzyme-linked immunosorbent assay. It is abbreviated as ELISA. Elisa or Elisa, both are correct. You should remember the full form of ELISA, which can be asked in MCQs. Enzyme-linked immunosorbent assay. We'll see meaning of this. So why it is called as enzyme-linked immunosorbent assay? So you know the enzymes are highly specific towards its substrate. And in this test, there is a use of the typical reaction between enzyme and its substrate. Okay, that is why enzyme. And it is linked immunosorbent. Immunosorbent. Immuno is antigen or antibody. Okay, antigen or antibody which is absorbed on the material specific for one of the components, and therefore it is called as immunosorbent. Assay is what? Assay is quantitative estimation of any substance is called as an assay. Okay, so therefore the word is enzyme linked immunosorbent assay. So in this, there is maybe the use of the variety of absorbing materials such as cellulose or agarose, or a solid phase such as polystyrene, polyvinyl or polycarbonate tubes or microvelles, or membranes or discs of polyacrylamide, paper or plastic. ELISA test is usually done by using 96 well microtiter plates, right? These are suitable for automation. We'll see the different types. There are different types of ELISA tests. Okay, we'll see. We'll go further little as yes, this is the diagrammatic presentation of three different types of ELISA, such as sandwich ELISA, indirect ELISA, and competitive ELISA, wherein there is a slight difference between all the three tests with respect to the way in which it is carried out, with respect to the component with respect to the antibodies, the way antibodies is used, the way antigen is used, and that is why the name is given like this. Look at the first sandwich ELISA. You will find that this blue colored are antigen. Antigen gets sandwiched between the two antibodies, and therefore it is called as a sandwich ELISA. Okay, so we'll see this in detail. So in this sandwich ELISA test, which is generally used for detection of rotavirus, the rotavirus is responsible for causing dysentery and diarrhea. So the sample used is a fecal matter. The various of a microtiter plates, right, are coated with a goat anti-rotavirus antibody. After thorough washing, that is, one is antibody. Now the fecal sample to be tested is added. Okay, after thorough washing. Why the washing is done? So that unadsorbed molecules will be removed out, and therefore washing is done. Then it is incubated overnight at 4 degrees centigrade for 2 hours at 37 degrees or at 37 degrees centigrade for 2 hours. Then the suitable positive and negative controls, they are also set up along with the test. Whales are washed again, and guinea pig anti-rotavirus antiserum. That is the second antibody. Now first antibody, then antigen, and a second antibody. This second antibody is labeled with alkaline phosphatase. This is an enzyme. Okay, as the name indicates, enzyme linked immunosorbent assay. Enzyme is linked to what? Enzyme is linked or labeled to the antiserum, and that is antibody. Antiserum contains antibodies added and incubated at 37 degrees centigrade for 1 hour. After washing, a suitable substrate is added. So when the enzyme is added, we need to add the suitable substrate. Here, for alkaline phosphatase, the substrate used is para-nitrophenyl phosphate. You remember this for MCQ. For alkaline phosphatase enzyme, the substrate used is para-nitrophenyl phosphate. It is added, held at room temperature along with the positive control, right? And it is observed till it gives the yellow color, just as in a positive control. The enzyme phosphatase splits the substrate to give the yellow colored compound. If the test sample contains rotavirus, it is fixed to the antibody coating wells, and when the enzyme labeled antibody is added, it is again in turn fixed, and the presence of residual enzyme activity is indicated by the development of yellow color. Yellow color indicates that the test is positive. So there is one more way by which the intensity of the yellow color is measured with the help of ELISA reader. It is called as ELISA reader, right? Which reads or which detects the intensity of yellow color and which gives indirectly the amount or number or concentration or titer of the antibodies present or antigen present in the clinical sample. So as just now we have said that if the test is positive, it gives yellow color. If the test is negative, there is no significant change in the color. Okay. See, in the indirect ELISA, what is happening is antigen is there. These are the antibodies. Okay. This is conjugate. Conjugate is what? Antibody to antibody. Conjugate is what? Antibody to antibody. Okay. Look at this. This is the antibody and this is antibody to antibody which is added. Okay. That is called as anti-gamma globulin. And then which is labeled with an enzyme. Okay. This conjugate, why it is called as conjugate? Antibody to antibody which is labeled with an enzyme, and then the substrate is added to which gives a positive reaction. That is the color change is there. So it indicates the positive reaction. Okay. We'll see this. So in indirect ELISA, detection of antibody by ELISA can be done. Example is anti-HIV antibody test, which is used for screening of AIDS patients. ELISA is specially used for screening of, I'm using the word as screening and not diagnosis, screening of AIDS patients, in which the purified inactivated HIV antigen is adsorbed on microassay plate wells. Test serum diluted in a buffer is added to the well and incubated at 37 degrees centigrade for 30 minutes. And then wells are again washed thoroughly. And if the serum contains anti-HIV antibodies, it will form a stable complex with HIV antigen. Right? Just I have shown in the diagram. Then anti-human immunoglobulins, that is anti-gamma globulin antibody conjugated with horseradish peroxidase. Horseradish peroxidase is an enzyme that is conjugated with the anti-gamma globulins. Then again incubated for 30 minutes. And after thorough washing, the substrate orthophenylene diamine dihydrochloride is added. So you remember for MCQ the name of the enzyme and its substrate. Here for the HIV testing, horseradish peroxidase enzyme is used, and for which the substrate is orthophenylene diamine dihydrochloride. Again it is allowed to react for 30 minutes. If there is development of color, it indicates that the test is positive. Okay. But along with every test, we have to always keep a positive and negative control. In a competitive ELISA, as the name indicates, competitive ELISA uses both unknown antigen, that is a sample, and a known antigen, that is a standard antigen, that is known to us, right? Which are allowed to compete with each other for the fixed amount of antibody. The amount of antibody is fixed, but then we don't know the nature of antigen in this case. So unknown antigen is, that is a patient sample, clinical sample is used, and the known antigen, that is called as, whatever is known, that is called as the standard. So competitive ELISA yields, again gives the inverse curve, wherein high values of antigen in the sample gives the lower amount of color change. Okay. So that is but natural, because it is the competition of binding with the antigens. The amount of antibody is fixed, but there are two different antigens. One is known antigen and the another is unknown antigen. So basically this test as competitive ELISA is used for detection of heptanes. Heptanes are incomplete antigens. Okay. What are the uses of ELISA? ELISA is very, very significant, remember, and plays a very important role in diagnosis of variety of different diseases. It is used for detection of HIV, used for detection of infectious diseases like hepatitis, right? Epstein-Barr virus, then cytomegalovirus. It is used for detection of dengue, influenza, then torch panel, used for detection of rotavirus in fecal samples, detection of enterotoxin of E. coli in feces, used for detection of syphilis or Haemophilus pylori IgG antibodies or antigen detection also. It is also used for detection of food toxins like chloramphenicol, streptomycin, penicillin, aflatoxins, which are food toxins, and food poisoning can be detected through this. Food adulterants, including E. coli, Campylobacter, Salmonella antigens can be detected with ELISA. Then mycobacterial antibody detection in tuberculosis can be done with ELISA. Whereas human allergen-specific IgE and IgA are detected with the help of ELISA. The advantages of ELISA are, it is a versatile test, it is highly sensitive, extremely simple, extremely minute quantity of the sample is required, so that it can be carried. Nowadays, this ELISA is carried out at the field, right? Because especially for the HIV testing, the person is not willing to come to the laboratory and carry out the test. So in such a cases, ELISA test can be carried out at the field. That means at the person's place. Just we have to take the blood sample and we have to carry out the test. It is highly economic as compared to other tests, and there is no we can say hazard of radiations. Okay, and therefore the test is most commonly used and most popular test for diagnosis of variety of different diseases.